Hi everybody. So welcome to my channel. So this is the second video that I'm uploading from the lecture series. Okay. So this is this lecture series which is there. This is dedicated for anybody who want to learn up more about life sciences. So like biotechnology, genetics, and biochemistry. So here in this lecture series, we'll discuss all the mechanisms or pathways or important crucial biochemical processes that happens in your cells, okay, which is very important for the healthy functioning, okay, maintaining healthy, uh, maintaining the health of that organism, right? So it's very important, and so like a and these are okay, it's very crucial, okay, crucial processes we'll be discussing, and we'll discuss much more aspects, many more topics regarding, we'll discuss a little bit of microbiology, all that. So here, so in the last previous video, we had discussed about P53. In this video, we'll discuss on apoptosis. So apoptosis is, what is apoptosis? So it's programmed cell death. I think everybody knows that. So why is programmed cell death so important? Okay, its purpose, its mechanism, its regulation. We'll discuss on the why it is important, okay, the in protecting your body, right? Why program cell death is so important. So we'll discuss all that, right? So we'll discuss also some of the proteins and enzymes that are involved in this as well. So let's begin. Now first we have to know, so most cell deaths in animals, they happen through program cell death, okay. So this is a carefully regulated fate of, okay, that decides the fate of your individual cells in an organism as whole, okay. So so program cell death is so important because it makes sure that anything unwanted or not harmful are removed, any harmful like DNA damage or any kind of stress, anything like that, it is removed. It makes sure any cells with such kind of, they are all removed, okay. So unnecessary cells are, okay, all these are removed, okay, that is why it's important. So programmed cell death is an active process, that is it depends on energy, okay. So it's usually there's a series of cellular changes that happens, the steps of cellular changes that happens, that is what is apoptosis is, that is programmed cell death. So there are morphological and biochemical changes that happen, okay. So apoptosis is a genetically controlled process, that is it deliberately, it's a process where cells deliberately self destruct itself, okay. So that and they do it through a series of regulated, very much regulated steps, okay, and they're done by certain enzymes called as caspases. So caspases are like a proteases, they're a class of proteases, so they help in this activation and in the cell death and all that. So we'll discuss this now. Okay, so so this process, why it is very crucial, because it makes sure that any unnecessary, damaged, or any potentially harmful cells, they are removed from an organism in a controlled manner. So it makes sure that is the tissue is healthy and it's functioning. Okay, then it's important. Okay, another. So why do you need cell death, or why do you need apoptosis? Why is this so important? So first it's required for balancing your cell proliferation and maintaining the constant cell numbers in tissues when it undergoes cell turnover. So a great example is that, so around 5 to 10 to the power 11 blood cells, they are eliminated daily in humans through programmed cell death. So this makes sure there is a balanced production that is there in the bone marrow, okay, and it provides a defense mechanism where the dangerous and damaged cells, they are removed, okay. So like virus infected cells, okay, they are frequently, they undergo programmed cell death, so this prevents the production of new virus particles, and they will make sure that the virus doesn't spread through your host organism. Another important is DNA damage. So DNA damage also induces programmed cell death. Okay, it makes sure that it eliminates cells which have any harmful mutations, so that will prevent from development of cancer. Now, also it's important for development. Development in the sense it's important for tissues. Okay, it eliminates variety of tissues like larval tissue that is there for amphibians and insects in their metamorphosis stage. It eliminates tissue that is there between the digits, that is there in your fingers and toes that are there. Okay, it helps in the development of mammalian nervous system. So it helps in so many of the neurons, right, when they develop, many of them get they undergo programmed cell death. Okay, they die through apoptosis. But the ones that are surviving, so they survive based on the ability to it acquire some kind of growth factors or survival factors. It should have these factors. So the cells which have these factors, those neurons only will survive, and they will go on to become like you know make your nervous system, right? So that's also it's important. It apoptosis or programmed cell death has a role in the association, regulating associations between cells and tissues. Okay, now what are the key features? So apoptosis was first described in 1977. Okay, so during apoptosis, the chromosomal DNA, it gets fragmented because of the cleavage that happens between nucleosomes, and the chromatin will be condensed and the nucleus breaks into pieces, small pieces, and these pieces, so the cell will start shrinking itself and it will break up into membrane enclosed fragments. So these fragments are called as apoptotic bodies. Okay, so your apoptotic bodies and cell fragments, they are removed through phagocytosis. Okay, that is done by your macrophages and all. So how is this done? How these cells are okay, apoptosis. So cells that die through this apoptosis are removed rapidly. How they are removed rapidly? So there is a signal that is given in the cell surface. Okay, so that signal will mediate that expression will be there. So that will tell you to remove these apoptic cells. Okay, these cells that have you know been eliminated through apoptosis. So it is done through phosphatidylserine, which will be present in the inner leaflet of the membrane. It will be previously it will be in the okay, so that phosphatidylserine will make sure it will give you like the signal, okay, for to remove these apoptic cells or cells that have died through apoptosis, so they will be removed. Okay, so yeah. Now, okay, so now next we should know what is the difference between necrosis, okay, that is, and necroptosis, because these are two terms, two other cell deaths that happens in us, in our cells, okay. So necrosis is basically it occurs due to acute external injury, that is due to any kind of toxins. Okay, if you are exposed to any toxins or something like that, that means externally. Okay, so they are uncontrolled and they are passive. Okay, and they are like and one of the thing is that the cells will start swelling up, and there is rupture of your membranes. Okay, there is unregulated release of cellular contents, and these like will burst into your surrounding tissue and they can spread. So they can go into other tissues as well. Okay, so this will trigger inflammation and all that. Necrosis affects group of cells, okay, not just one cell. And because of that, there is chances of swelling. You have swelling and pain and redness and all that. So necrosis, this is more, you will see with respect to if you're like toxins, like any with skin kind of issues, skin in toxicology and skin toxins. If you discuss skin toxicity and all, right, you will hear necrosis more. Necrosis is more like it's bad, it's a little deadly, it is a little harmful as well. Okay, necroptosis is basically it's this happens when this is a, like as I said, backup cell death pathway. So if the apoptosis is blocked, okay, so they can be blocked by your caspase enzyme, right? If that is blocked, okay, then if your apoptosis is blocked, then your necroptosis is the backup cell. It's initiated. So this is regulated and it is programmed. So, but the cell dies through a necrosis kind of way. That is, the cell will swell up and the membrane will rupture, Okay, and the contents will be, so this is triggered by TNF. TNF means tumor necrosis factor. Okay, we'll discuss TNF also in the, so this there is alpha binding that happens to the receptor, so they initiate this backup cell death pathway. Okay, uh, okay, now next we'll discuss. Okay, now next we'll discuss the pathway. Now we'll discuss on the mechanism of apoptosis. Now how apoptosis is programmed cell death, that there are two activation pathways that happens in apoptosis. So you have extrinsic, intrinsic, okay? And there is mitochondrial involvement and there is caspase activation. And at last there is that execution, that is cell death. So how it happens? First, intrinsic pathway. Intrinsic pathway happens due to DNA damage. Okay, that is because of a lack of growth factors or survival factors. Okay, so it involves mitochondria. It's mitochondria dependent. It depends on mitochondria. It occurs in the mitochondria. Most of this process. Okay, most of this pathway and the initiation and all this stuff. Okay, happens in mitochondria. That is why there is a involvement in mitochondria. So the cellular stress, whenever this cell have any kind of stress, okay, or the DNA is damaged, for example, then it will activate certain proteins which are called pro-apoptic family proteins. Pro-apoptic are these genes or proteins. Okay, these are the genes that initiate or they promote apoptosis. Okay, so they will okay, so they will okay, so they will so this you have this BCL2 family proteins. Okay, these BCL2 family proteins, there are a range of family proteins. Okay, now they will go and they will activate these proteins like BAX and BAC. Okay, that are pro-apoptic. Okay, so BCL2 we'll discuss in the later. This BCL2 family, they're more like a they mostly most cases they inhibit, or they what to say, they stop the apoptic process. But the stress, any kind of stress, if the cell is in, if the cell faces any stress, then this is activated. This particular BCL2 family proteins activated. Okay, so they will activate this pro-apoptic family proteins. Okay, backs and back. So the proteins will go and migrate to the mitochondrial outer membrane, and they form pores. Okay, so the pores, and it will give you this mitochondrial outer permeabilization. That is, it's called MOP. What is that? So this MOP, it will cause, so that is the outside. These pores will go these are formed in your outer membrane of the mitochondria. So that is, it's called mitochondria outer permeabilization. So the changes happens in the membrane. Okay. So that causes a release of pro-apoptic factors from the mitochondria inner membrane. It will be released into the cytosol. So that is one such important factor, pro-apoptic factors. That's what it is. Pro-apoptic factors, or these are stuff that initiate apoptosis. Okay. We will discuss on BCL2 family protein in the upcoming slide. So you will get to know more. So they will release cytochrome C. Okay. So we all know cytochrome C are present in mitochondria, right? That's why mitochondria is involved. That's the importance of mitochondria. So this whole pathway of apoptosis actually kind of occurs in your mitochondria. So it's so important. So this is how basically your intrinsic pathway happens. Okay. Now extrinsic pathway, it involves any kind of extracellular signals from any other cells outside. Now this extrinsic pathway involves some kind of death receptors, and which is there on the cell surface. Okay, they involve death receptors that are called as TNF. We have discussed before, tumor necrosis factor receptor superfamily. Okay, and that okay, and they will be having, and they have specific kind of, that includes some like FAS. Okay, FAS is one such death receptor, and you have another called TNFR1. Okay. Now, and you also have certain ligands. So since you have a death receptor, then you will need some kind of ligand. So you have a death ligand associated with it. So you have FAS-L or TNF alpha. These are such death ligands. So these ligands will go and bind to these death receptors. Okay, they will be very specific. Okay. So there is a formation. So this binding will, when the ligand will bind to a receptor, it will cause some change happening in the, okay, changes will happen, structural changes happen that will recruit certain proteins called adapter proteins. Okay, so adapter proteins, example of that is FAD, FADD. Okay, and they will enact, and this will enact. So they're an inactive pro-caspase. Okay, the so now we are coming into your caspase. So we will discuss. Okay, so inactive caspase 8. Okay, so that will be, and that will then there is a, what is it, there is, so this whole thing will, this will form like a complex, this whole, ligand interaction, ligand and the receptor interaction, along with sorry, along with the change that has happening in the protein, in your complex, the structure change, along with this adaptive protein and your inactive procaspase, this will you'll get a death inducing signaling complex it forms. This is called disc complex. Okay, now this then there is the self cleavage happening, then there is self cleavage and activation happening. Okay, that will activate caspase 8 initiator. Okay, caspase 8. Okay, so this is very important because caspase 8 is an important initiator caspase. That means these are the caspases that initiate the subsequent processes in cell death. Okay, they initiate. They are what you call. They are the ones who will activate the other downstream. So there are many. So there are various caspases. Caspases. This is a family of. It's a family of proteins. Okay. So there are so you have caspase 8, 9, 10, 7, 3 like that. Okay. So certain caspases are they are like initiator caspases categorized. They will initiate. They'll be they will active. They are activated and they will initiate. Okay, initiate the activation, okay, of subsequent caspases. So those subsequent caspases are known as effector caspases or executor caspases. These are the ones that will, their example is caspase 3 and all. They initiate, these are the ones who will go and initiate, your cell death. Okay, so yeah, so caspases are very important. Okay, so again coming to your mitochondrial, so mitochondrial involvement. Okay, one crucial, the BCL2 family. Okay, this BCL2 family, they are actually they are present in your mitochondria. Okay, so so when you activate your pro-apoptic effector proteins, okay, effector proteins, that is your bags and back. Okay, they will oligomerize, they will change some shape, will happen, they'll form pores. Okay, that those pores will happen in the outer membrane, and there is a release of cytochrome C. Okay, so the release of cytochrome C, so I am in this this mitochondrial involvement I am discussing here. So this there is a release happening of cytochrome C. So the cytochrome C will go on and activate your caspases. Okay, so in your extrinsic path, okay, so in the intrinsic pathway, one of the main caspases are caspase 9. So caspase 9 is activated. It forms complex, okay, which is called as APF1. Okay, APF1. It forms, and that will form like a multi-subunit, which will result in apoptosis. Okay, apoptosis. Now, what happens now? This caspase 9, it will cleave the and it will activate your downstream caspases, effector caspases, as I told before, caspase 3. Okay, that will result in cell death. Okay, so okay, now if you, so this is the intrinsic. I have just given a gist of intrinsic. So let us just recap whatever we discussed in intrinsic. Okay, just to give you a thing. So in intrinsic, there is first you is activated through any stress or damage. Okay, some kind of stress, DNA damage or something. And then after the okay, after it has been okay. So after that, what happens? Then you will have it will activate the BCL2 family proteins. Your BCL2 okay, they will go and activate pro-apoptic, pro-apoptic factors or effector proteins. Okay, like BAX and BAC. BAX and BAC is activated, which are pro-apoptic proteins which initiate apoptosis. Okay, now then the proteins will go and go to your mitochondria. Okay, and they will mitochondria outer membrane and they form pores, which are known as MOMP, which is mitochondrial outer polymerization okay membrane. Okay, so they are okay, you can say outer. Okay, so this is the thing. So this will cause your the release of cytochrome C. Okay, so these factors are present in these factors, that is your pro-apoptic factor. Okay, they are there in your outer membrane. Now they'll be released. So from okay, those pores that will so this MOP, the spores which are there, the factors are released from the intermitochondrial membrane. Okay, there'll be that's intermembrane space which is there, right? That will be released into the cytochrome C. Okay, okay. Now, okay, they will be released. Okay, so now that is the intrinsic pathway. So okay, and after that there is this your okay, so so when your BCL2 family, okay, they are involved. So these BCL2 family are involved in this apoptosis. They are crucial family of proteins. Okay, so these guys, they will go and activate pro-apoptic effector proteins, which are known as BACs and BAC. So there is change in the shape, change in the shape, and uh, that leads to the pores pores which are there. That is your MOP pores. That will be present in the outer membrane. Okay, and the cytochrome C is released. Okay, so cytochrome C is released from the intermembrane space in the cytosol. Okay, it is released. Now, the release of cytochrome C, it will initiate caspase activation. The in your intrinsic pathway, the key caspase is caspase 9, which is your initiator. Okay, this initiator caspase 9 will go and form complex, which is known as APAF1 multi-subunit, and that complex is called apoptosis. So then this caspase 9 gets cleaved. It cleaves and it will activate the downstream effector caspase, which is caspase 3, and that there is cell death. Now coming on to the extrinsic, as I told you, there is involvement of death ligands and death receptors, and you have the change in the shape there. Okay, so okay, so there you have, so there is this TNF, which I have told before, the TNF, which is your tumor necrosis factor. These are your main component of ligands and receptor. Okay, so these are this is one of the main like they form like identical, like polypeptide chains with another three identical polypeptides. They form like a chain. Okay, your TNF family, TNF, and there is and there is a binding of they'll go and bind these chains, these polypeptides will go and bind to these specific receptors, and they'll cause changes. Okay, that's a trimerization, changes in the shape. Okay, and then then these receptors will go and bind to adapter molecules. One example is FADD. Okay, so these adapter molecules will go bind to caspase 8. Caspase 8 is the important initiator caspase for extrinsic pathway, and they'll go again and activate okay, they'll activate other downstream effector caspase like caspase 8, caspase 3. Now this is one way. Another thing is that if there is sometimes you need amplification of signal. Okay, in the case of extrinsic pathway. In that case, the caspase 8 cleavage, it will go and cleavage. BID is a pro-apoptic regulatory protein. It will be cleaved. Okay, and this BID, which is that PID, this will result in the permeabilization of mitochondria, and that results in the activation of caspase. Okay, caspase 9, caspase, and there is there is caspase, and there is the it will amplify your caspase cascade is amplified. It is initiated, so that results in the activation, direct activation of caspase 8 at the cell death receptors. Okay. Now, what are caspases actually? Caspases are important executors of apoptosis. These are the, as you see, so it is these uh, okay, these caspases, which are the specialized enzymes that actually do the you know no, cell death, the process of initiates the mechanism or pathway of apoptosis. So they are proteases essentially. They are they have cysteine residue at their active site, and they cleave after the aspartic substitute proteins. Okay, so caspases are the ultimate effectors or the executors of cell death. Okay, yeah. So yeah, now next we'll go on to the genes that regulate apoptosis. So there are one is pro-apoptic genes. You have anti-apoptic genes. You have tumor suppressor genes and oncogenes, and then finally there are gene mutation effects. So pro-apoptic genes are the genes that promote apoptosis. So that example is back. Okay, so these backs and back they trigger mitochondrial permeabilization. Okay, they help in the release of cytochrome C. Okay, cytochrome C, they activate the caspase cascade. Okay, and they will result in the cell death. Okay, so then there is anti-apoptic genes. This involves one main is your BCL2 family proteins. Okay, so these are the genes that prevent apoptosis. So they help in the promotion of cell survival. So for any like neurons and all, right? So these guys, this family, there is BCL2 and there is BCLX XL. Okay, these are the two families. Proteins that are there in main proteins that are there in this family. So they help in stabilizing what do you say, the mitochondrial membrane, and they stop apoptic factors, so they block it. Okay, so this is it prevents overexpression. Now how does your BCL2 family proteins, how do they interact with your, now we have seen in the previous, is the BCL2 family proteins that interact with your backs and back and they trigger apoptosis. How does it do that actually? So it does that. So BCL2 family proteins control it by interacting with pro-apoptic genes. They're okay. So backs and back is present at your mitochondria. Now, under normal conditions, the BCL2 family proteins, they will go and bind to backs and back, okay, and the family proteins, okay, they will initiate, it will be active here, backs and back, right, and that will, okay, it will bind to these backs and back, it will make it inactive, it will become inactive, and that will prevent the mitochondrial outer membrane from permeabilization, or that is forming pores and the subsequent release of cytochrome C and all that. But if there is any kind of stress, then another set of BCL2 family proteins, you have BH3. BH3 is this BH3 only proteins you have. Okay, these are the proteins. This is also part of this BCL2 family. They'll go and they'll be activated. Okay, then the backs and back. Okay, they backs and back. Okay, so they either will go directly and bind to backs and back, and they'll activate, or they'll bind to anti-apoptic BCL2 family proteins as well. They'll go there and bind, and they will displace the backs and back. There is change of the shape will happen. Okay, then there is they'll insert in the mitochondrial pores. Okay, and then there is mitochondria will permeabilize. Okay, the outer membrane will be permeabilized, and then the cytochrome C is released. Then there is the downstream caspase activity, caspase cascade, and there is the cell death. Okay. Now, another important genes are your tumor suppressors. We have discussed this in the previous, right? How p53 is actually an important activator of your apoptosis. So it activates it whenever there is a damage in your DNA or there is a stress. So they'll activate this pro-apoptic genes which you have discussed, right? This backs and back. They'll go initiate them, and they initiate apoptosis. So okay, so there is that will prevent tumor. Then the another is oncogenes. Oncogenes, one example is CMYC. So these can promote cell survival. Okay, oncogenes and growth and proliferation. But when these genes are mutated, okay, then there is a disruption happening in apoptosis, and that will lead to cancer. So what is the CMYC? These are genes that is oncogene that produces a transcription factor that is important for cell regulating cell growth, proliferation, metabolism, and apoptosis. Okay, this is important. Okay, when this gets mutated, you get cancer. Okay. Now, gene mutation affects. So, if any apoptosis is affected, it will the regulation. If the okay, like gene mutations, if they are happening, they will blocking. If there's any blockage happening to apoptosis through any kind of mutations, it affects the pathway. It affects this process of apoptosis. So, any mutation, if it happens in your tumor suppressor genes, they will impair the apoptosis. Okay, and and your damaged cells will survive, and then you get cancer. So it will develop into cancerous cells. Okay, so and the overexpression, right? Overexpression of anti-apoptic genes. So that will result in resistant to cell death. Okay, so that is one reason, main reason how this cancers those survive. They apoptosis cannot kill it. It's not affected by apoptosis. This is one of the main reasons. So so if the apoptosis, the regulation of apoptosis is so important. Why? Because if there is a limitation of apoptosis, okay, very little apoptosis happening, then you can have cancer. And if there are overexpression or more number of apoptosis happening, okay, then what will happen? It results in degenerative diseases like Alzheimer's and Parkinson's. One of the main causes is this overexpression of apoptic cells. Okay, because it means that there is more cell death happening, right? Overexpression. So the genes that are important or factors important for growth, right? All that can get severely affected. Okay, so that's a okay. Now, what are the important applications when it comes to apoptosis? So, apoptosis helps in role. They have role in development and immunity. How? They help in shaping your organs. Okay, when during your embryonic development, they're important in removing autoreactive immune cells. So, cells that immune cells should not attack your own self cells, right? So they remove any cells, if any cells that attack its own immune cells, right? Your own immune cells, so those cells are removed through apoptosis. That's why again it's important in the development. Okay, it's again important for autoimmunity basically. Okay, it is important. So if it's in the involvement of apoptosis in diseases. So dysregulated apoptosis results in diseases. Okay, as I told you, if there is less apoptosis happening, you can have the cancer, and if there is more, then you can have degenerative diseases. You can have immune uh, diseases or diseases relating to autoimmunity problems you can have. Okay, so then we have the therapeutic targeting. Okay, so what cancer therapies, okay, are important. We need therapies that can reduce apoptosis. So in the case of Alzheimer's, cancer therapies for inducing apoptosis in the case of cancer. Okay, and finally, so why it is important in research. Okay. Now the mechanism of apoptosis, it's very important to know about the cell regulation, mechanism of cell regulation development. Okay, and it's important for treatments, that is for cancer and autoimmunity and for degenerative disease. It's important, it helps in understanding developmental biology and immune system. So this is why apoptosis is very important. Okay, so yeah, so these are the summary of it. Okay, main things that have been covered. So I hope you guys all like this second video of the lecture series. So there will be more coming. Okay, so I hope you guys liked it. So thank you.